All right, let's talk about water conservation. To give you a sense of how important this is, the amount of filtrate that is produced, or the amount of fluid that comes out of the glomerulus and is collected by the glomerular capsule passing through the tubule every day, ranges from 120 to 180 liters in the average adult body. That's many times more than the actual volume of blood that we have, which is 4 to 6 liters. And that means that the blood is passing through and getting filtered multiple times every day. At each time, all of the useful substances and water come out, and the useful substances and the water have to be moved back in. Now, the vast majority of the water that comes out in the filtrate is reabsorbed by osmosis. So as the proximal convoluted tubule transports glucose back in, for instance, or sodium back in, water follows those particles and is reabsorbed by the body. However, some of that water is going to stay by osmosis and follow the nitrogenous wastes that we're trying to get rid of. That amounts to about 30 liters, which is still more than the amount of blood that you have in your body at any one given time. And if you imagine trying to urinate 30 liters a day and drink 30 liters a day to replace it, you start to get the sense of why water conservation is such an important process in the kidneys. Now, how do we do this water conservation? When we talked about the structure of the nephron initially, I mentioned that in juxtamedullary nephrons, the ones that have the really long nephron loop that goes into the medulla, that the nephron loop is responsible for making the medulla salty. So part of what's required is for this nephron loop to build a salt trap. So all of this space down here is going to be made salty. The other part of what we need in order to conserve the water is a tubule that's passing through that is permeable to water. Now, this tubule is permeable to water and is under hormonal control, which means that we can determine how much water needs to be conserved at any given moment in time. So when we're dehydrated, this will conserve more water or allow more water to come back into the body. If we are adequately or overhydrated, then it will allow the water to stay in the tubule and go into the urine. So first, let's talk about how we make the medulla salty, and then we'll talk about how the collecting duct works to let the correct amount of water go back into the body and follow that salt trap. So the nephron loop makes the medulla salty through a mechanism known as countercurrent multiplication. It's a classic positive feedback loop, but not the bad kind. This one is just constantly making the medulla salty, or constantly adding salts to the medulla. Now, let's take a moment and look at the diagram first before we go through the steps. This is the proximal convoluted tubule. This is the nephron loop, and this is the distal convoluted tubule. If you look at the limbs of the nephron loop, you'll see that the one that is bringing the fluid down, which is called the descending limb, is thin. And that means that the cells here are permeable to water, but not to salts. The ascending limb of the nephron loop is thick, and that means that the cells there have salt transporters in them and are permeable to salt, but not to water. So those are the two key features of the nephron loop that allow it to make the medulla salty. The other thing I want to point out here is that there are numbers and a color gradient inside the nephron loop to indicate how concentrated the fluid is inside. These numbers are the measurement of osmolarity, or the number of particles that could attract water inside the tubule. So 300 here is actually pretty dilute. It's more water than it is salts. But you'll notice that as we move down through the tubule, the numbers get higher. That means that the salt concentration or the osmolarity is increasing. And the artist has also made it darker and darker in order to indicate that the fluid inside is becoming more concentrated with salt. As we move back up in the ascending limb, you'll see that the numbers get lower, and the fluid that's leaving the nephron loop is actually more dilute or has more water in it compared to salts than the fluid that was coming in the first place. That indicates that it's leaving salts behind, and what's staying in the nephron loop is the water component. So the fluid that's arriving at the nephron loop from the proximal convoluted tubule is actually pretty dilute. It has a high water concentration. This is because the proximal convoluted tubule has been taking substances out of the tubular fluid to send them back to the blood, where they're picked up by the peritubular capillaries. And while water follows, water doesn't go immediately with it. So there's sort of a lag. So some of the water is left behind, making this fluid somewhat more dilute. Because this fluid is dilute, when it passes into the descending limb, which is permeable to water and not to salts, the water starts to leave the tubule and move towards the, we're going to say that it's saltier, but it's not much saltier yet because we've just started this process. All the same, there's more salt out here, so water leaves the tubule, and the fluid that's inside the tubule becomes more and more concentrated or saltier because the water has been leaving. And salts can't leak out of this limb. By the time the fluid reaches the bottom of the nephron loop, it's quite concentrated. And as it starts to move up through the ascending limb, the salty fluid inside the tubule stimulates the salt transporters in those cells to pump out salts. The saltier the fluid in here is, the more those transporters pump the salts out. Again, working with a gradient. The most salt is pumped out down here where the fluid is the most concentrated. And that means that we're also getting more salt at this part of the medulla, and the medulla up here is less salty. So the deeper we go, the saltier the medulla is. All right, because we've been pumping out all the salts, the fluid becomes increasingly more dilute or less concentrated, and leaves the nephron loop and goes into the distal convoluted tubule. The next batch of tubular fluid that's coming in is also dilute. And so as it reaches the descending limb, which is permeable to water, because it is dilute and because we've already added some salts out here because of the salt transporters in the thick ascending limb. So now this is even saltier. That means more water is going to leave. The more water that leaves, the more concentrated this fluid becomes. The more concentrated this fluid becomes, the more salts it pumps out, and the saltier the medulla gets. The more salt it pumps out, the more dilute this fluid becomes. Next batch of fluid comes in, same thing, it's dilute. Because the medulla is even saltier than it was before, even more water leaves. That makes this fluid even more concentrated. Because it's even more concentrated, we put even more salts inside. So this is a mechanism because we have one side that's permeable to water, one side that's permeable to salts, because they move in opposite directions. We end up with a system to leave the salts behind and just carry the water away in the tubular fluid. Now, some of the water that is that leaves the nephron loop, you would think that it would just cancel the effect of the salts that are being left behind. But that's where the vasorecta, the specialized vessels that we looked at with nephron structure, come in. I'm not going to go into all the mechanisms here, but the vasorecta, or the straight vessels, which surround these special nephron loops, because they are arranged with the flow of blood in that's coming into the vasorecta going one direction and out the other, and they're parallel with one another and parallel with the nephron loop, the blood that enters the vasorecta, and remember, it's here to conduct exchange and drop off oxygen as well. We have to send those vessels there. But they are permeable to both water and salt. So the blood traveling through the vasorecta on its way to deliver oxygen is becoming increasingly salty as it goes down. And that's because there's a gradient. It's salty in the medulla, so the salt goes into the vasorecta, but so does water. And as the blood continues to travel and reaches the bottom of the vasorecta, it's almost as salty as the medulla is, the surrounding medulla is. I can't speak. Now, when it turns around and goes the other direction, as the blood inside is super salty, it matches the saltiest part of the medulla. As it moves this direction, the salty blood, there's a gradient for the salt to leave the blood and go back into the medulla, because we're moving back up where it's becoming less concentrated in the medulla or less salty in the medulla as we move back up. So that causes the salts to leave, but the water largely remains behind and stays in the circulation. So these vessels provide a mechanism for delivering oxygen, picking up carbon dioxide, and passively taking on the salts and letting them go, simply because of how they're arranged in a parallel structure. Okay, so now we have a sense of how the medulla is made salty by the nephron loop. Let's look at the collecting duct to see how the water conservation works. Now, because the medulla here is super salty, any water that is coming through the collecting duct can be allowed to leave the collecting duct through these special channels called aquaporins. So water leaves the aquaporins and goes towards the salt. The vasorecta are over here to pick up the water. That means that the fluid, which is becoming urine as it passes through, is becoming more and more concentrated. So the more of these channels that we have here, the more water is allowed to go into this space and be picked up and brought back into the bloodstream. Now, interestingly, the cells of the collecting duct are hormonally controlled. The hormone is called antidiuretic hormone. Let's take a look at that word for a second, even though we've seen the hormone before. A diuretic is something that causes you to produce urine or increase the urine volume. So an antidiuretic hormone would be something that causes you to produce less urine, which means that antidiuretic hormone, when it is secreted, will increase the number of channels that are in this collecting duct and allow us to reabsorb this fluid or reabsorb the water before it reaches the urine, where we can't adjust the composition anymore. If there is less antidiuretic hormone secreted, then these cells will have fewer channels. They actually put the channels in the membrane and take them out of the membrane in response to the level of the hormone. One of the things that inhibits antidiuretic hormone is the consumption of alcohol. So with the consumption of alcohol, antidiuretic hormone concentration decreases, which means that the cells of the collecting duct have fewer of these aquaporins in their membranes. Therefore, as this dilute fluid enters the collecting duct which is surrounded by this salt trap, there are fewer ways for the water to go towards the salt trap. In fact, the water is essentially trapped inside the collecting duct and added to the urine. So as you drink alcohol, your urine becomes more dilute and your blood becomes more concentrated, potentially resulting in you becoming dehydrated if you drink too much alcohol.